Welcome back to Human Biology with Dr. B. Today we talk about the senses, both the general and the special senses. So let's first talk about sensory receptors. Receptors are structures that are specialized to detect and respond changes both externally and internally. So your special senses sense external change and stimuli, and your brain translate that. And your internal receptors, they process information coming from the inside of the body and allow your brain and autonomic centers to address that situation. They respond by generating action potentials through their neurons, which we call electrochemical messages. And with the stimulus, if the stimulus, just like any other action potential, it needs to be strong enough in order to actually reach the brain. So then we get to talk about sensation versus perception, which are very, very different things. Now, sensation is awareness of a stimulus, so how it's experienced, sight, sound, etc. And the sensation depends on what type of the brain is processing that action potential. Perception is the conscious awareness of that sensation. So right now, my voice is causing a sensation, if you will, in your verbal processing centers in your temporal lobe, and you are able to translate. You then perceive, are aware of what I'm saying because your brain is able to take the sounds, take the vibrations, and process them into meaningful messages. And this occurs in the cerebral cortex, which integrates the sensory input. And here you can see this is how the brain processes it. So first we have a sensory receptor. This detects the stimuli. The neurons carry the impulse to the spinal cord and then to the brain where it's integrated. The brain then interprets, and based on the brain's interpretation of the understanding, an action is generated. Now, sensory adaptation is when your sensory receptors continue to send the message to the brain, but the brain, deeming other sensory input more important, does not make you aware of it. So sensory receptors stop responding when they're continuously stimulated, and this brings a decreased awareness of what's happening. So for example, if you were to sit quietly right now for a second, you'd be able to hear the whirring motor of your computer. Listen. There it is. Now, when you are listening to the video, what you hear is my voice, and what you see is the words and the pictures on the screen. All of this is processed. When your brain takes into account what's most important right now is what I'm saying and what you see on the screen, your brain, your thalamus, basically says, no, I don't need to process that information right now. I need to focus on the incoming sensory information that is of importance. And sensory adaptation it varies between any of the different receptors. Pressure and touch receptors adapt quickly, which is why after a few minutes of having pants on, you no longer feel your pants. You are still wearing pants, thankfully, because it is socially acceptable to be wearing pants. However, the touch and pressure of your pants that are putting on your skin, on the receptors throughout your legs, that is no longer processed by the brain. The brain is like, okay, I have pants on, we're good, and then moves on to the next stimulus. Receptors in the muscles and joints constantly report body position. These never adapt, which is really, really good because otherwise you'd lose track of your legs. Oh, I have a leg? Oh, okay, great. Now we have different classes of receptors. We have also classes of the senses. We have general senses, and then we have special senses. And the general senses, these are receptors are located in the skin, muscles, joints, bones, internal organs, all over. And they are mechanoreceptors, and we're going to talk more about each of these in a moment. Mechanoreceptors that sense a change in mechanical position, thermoreceptors that sense a change in temperature, photoreceptors that sense a change in light, chemoreceptors that sense a change in a specific chemical or ion, and pain receptors, which we also call nociceptors. And the special senses are limited to vision, your eyeballs, your hearing, your ears, your inner ear, the middle ear, equilibrium, which also is within your ear, smell, your nose, and taste, which is in your mouth. So the general senses, these can be anywhere from a free nerve ending to an encapsulated nerve ending that sends information on specifically where this is located to the brain and processes it. So one of the important things I want to delineate here is what's called a labeled line code. Now, as we move through each of these different types, we have free nerve endings, and these are specific for pain, heat, or cold. We can have free nerve endings wrapped around a hair that helps us process touch. We have the Merkel's discs. You can see the Merkel cell right here and the neuron attaching to it. This is light touch. This is more of a tickle sensation. And then we have Meissner's corpuscles that work with the Merkel's disc for light touch as well. We have Pacinian corpuscles, which process strong pressure, deep pressure, as well, like a pressing down. Think compression pants, and process that, as well as vibration, the vibrations in the car when you sit and turn on the engine. And then the Ruffini corpuscles, this is continuous pressure. Continuous pressure would be, at some point, probably processed as pain if it is too much. Now, you can see each of these have a neuron associated with it. And each of these neurons is specific for this type of sensation. So if you were to stimulate the Pacinian corpuscles with cold or hot, it would not process that. It would only process pressure because all of these neurons are labeled line code. So all of these neurons can only process the specific type of sensation that they are designed for. So Pacinian corpuscles, only strong pressure and vibration. So if you were to stimulate this with a cold or a hot stimulus, your brain would translate it as pressure. If you were to stimulate a Meissner's corpuscle with light touch, it would process properly. However, if you stuck a hot pin in this neuron, it would still process it as light touch. It would not process it as hot or cold. Now, if you were to put heavy pressure on free nerve endings, they would process it as hot, cold, or pain. Labeled line code. Mechanoreceptors are those that respond to touch, pressure, vibration, etc. They respond to stretching, compression, twisting, etc. These are primarily in the skin, and all of these various types of receptors in the skin are, because your skin is your largest organ, it's open to the public, it interfaces with everything in the environment, it's very important to keep that organ safe and healthy. This is an evolutionary trait. So, mechanoreceptors that detect touch, we just talked about Merkel's disc and Meissner's corpuscles, and they respond to various types of touching. Now, mechanoreceptors that detect pressure are the Pacinian corpuscles and the Ruffini corpuscles. Try saying that 10 times fast. Usually encapsulated nerve endings that are specific for this type of sensation. Thermoreceptors, they only respond anytime they are stimulated. They send information back to your brain about a temperature difference. So they respond to warmth as well as cold. And the hot and cold receptors are separate. So if you give a stimulus to a hot receptor with something that's cold, it will process in your brain still as hot if that's the type of stimulus you give it. Anytime a warm or a cold or a pain or a pressure receptor is stimulated, it will only process as hot or cold, pain or pressure. These also, the cold and the hot, the thermoreceptors, they adapt quickly, which means that once they're situated and we say, okay, we've sent the cold message to your brain, then all of a sudden it will turn off. Proprioception is done with muscle spindles within the muscle fiber. These are special muscle fibers that have specific nerve endings that help prevent overstretching or overcontraction or too deep of a contraction in the muscles. Because what happens if we overstretch the muscles? Well, they rip off the bone. What happens if we overcontract the muscles? Well, they rip off the bone. And so the tendon organs, the muscle spindles, these help measure and monitor the muscle tension to be able to protect all manner of movement within the body. They also work with information from the inner ear that allows balance and proprioception and understanding of where you are in space in order to protect the muscles and therefore the bones. Now, the eye, we're getting into the special senses. Do me a favor right now. Okay, you're going to think I'm crazy, but that's nothing new. So close your eyes and take a finger and put it on the eyelid of your closed eye. Now, very, very gently, I just need you to push on your eye. I'm not joking, push on it. Okay, now stop and think about what you just saw. You're like, but Dr. B, my eyes were closed. I didn't see anything. No, if you didn't see anything, do it again. Now, you are applying pressure to your eyeball, and your eyeball has nerve endings that are specific for photoreception. So what does that mean? They respond to light. So when you close your eyes and push on your eyeball, most likely you will see flashes of light because you are stimulating those receptors, and those receptors, labeled line code, go back to your brain and say, oh, hey, light, even though there is no light and your eyeballs are closed. It's the same reason that when you get hit in the head, all of a sudden you see stars because your the photoreceptors in your eye are still being stimulated by the pressure, by the movement that's going on. Yeah. So there are 3 layers of the eye. We have the sclera and the cornea on the outside, right here. The cornea is the clear part right over your pupil and your iris. And then the sclera is the white of the eye. The middle layer is the choroid and the ciliary body. The ciliary body is right here, and it is a muscle that attaches to the lens that changes shape. It changes the shape of the lens so that you can focus both near and far in different light settings. And then the choroid is this dark red colored internal portion. It's the same reason that movie theaters are dark red inside, because they focus the light onto the screen. The choroid allows the focus of the light through the pupil, through the lens, onto back here. The innermost layer is the retina. And the retina is a sensory layer. When you dissect the eye and you detach the retina, it literally looks like a bunch of snot. It's this gray, filmy thing. It's tissue paper, like it rips very easily. And that is the neurological layer that has the photoreceptors in it that senses and sends back to your brain the information on light. Now, there are focusing problems, and this has everything to do with the shape of the lens, as we talked about right here behind the pupil. We have farsightedness, nearsightedness, and astigmatism. We'll talk more about all 3 of these in lab next week. Causes can include discrepancies in the curvature and the thickness of the lens or the shape of the eyeball. This is typically called astigmatism. And with any of these things, normal vision can be restored through LASIK, or wearing contacts or wearing glasses, which most of us do at this point. Photoreceptors, when the light is focused through the pupil, through the lens, onto the retina, it causes chemical changes within the photosynthetic pigment, or not photosynthetic. We do not do photosynthesis, just so we're clear. They're photopigments in the photoreceptors that respond to the light, create a chemical change, and this is the stimulus that sends an action potential to the brain saying, hey, there's light. Photoreceptors release less inhibitory neurotransmitters, so it basically moves the neurons into a more excited state. And as the processing of the visual information increases, so does the activity increases the processing of the visual information. And then electrical signals from the retina go back through the optic nerve and are processed back here in the occipital lobe. You can see here are your eyeballs. They sit just under your brain. Your brain is directly over your eyeballs. And you have your optic nerve. It crosses over right here and projects to the opposite side of the brain in the visual cortex and the occipital lobe at the back of your brain. Now, hearing. Sound is so fascinating to me. In order to hear anything, the ear, this is why you have these awesome, they're called the pinna of your ear, that stick out, some of us more than others, on the side of your head. And their whole job is to collect and filter vibration, which is all that sound is, down into the outer, middle, and inner ear. Now, the loudness of the sound, is determined by the amplitude. If you look over here, we're talking in waves because this is how sound is projected. So low amplitude right here, low amplitude is soft. A higher amplitude is louder. Now, a lower frequency is a lower pitch, and a higher frequency occurs faster. You're welcome for all of that. And this is what the ear looks like. So we have the outer ear right here. We call this the pinna. External auditory canal. It goes all the way in here. Now, my anatomy and physiology professor in college said you shouldn't stick anything larger than your elbow in your ear. And if you think about that, you're like, oh, so maybe nothing? Yes, correct, nothing. Because your earwax is the cerumen. That's what's called. It's called the cerumen. It helps protect and clean the external auditory canal. The external auditory canal leads to the tympanic membrane, and the tympanic membrane vibrates with every sound that you hear, and it vibrates very similarly to a drum, and then it causes, once this vibrates, it causes the malleus, incus, and stapes to also vibrate. Now, if you've ever worn high heels on a very soft wood floor, you know that wearing flats on a hardwood floor wouldn't necessarily cause indentation. But when you wear stiletto heels on a hardwood floor, all of a sudden you're leaving these tiny little imprints of your heel on the floor. Why? Because the entire body weight is being pushed down onto this tiny little place. And so it increases the pressure because all of it is going down to this little tiny stiletto heel. The same principle is true here with the malleus, incus, and stapes. So we take the larger vibrations of the tympanic membrane, also the eardrum, and it is transferred to the malleus, incus, and stapes. See right here, this is where it's transferred. And then the stapes looks like a stirrup, and it pushes on the oval window of the cochlea. And the cochlea is right here. It's this snail-looking like thing. And this is actually inside, it resides inside the skull. And the cochlea is this snail-like thing, and inside it's this jelly substance with what we call ear hairs. Now, not ear hairs like old man ear hairs that stick out of the outer ear canal, and you're like, ooh, you should probably trim those. No, different ear hairs. These ear hairs function as transmitters. And so when the stapes pushes on the oval window, it causes waves throughout the cochlea. And within the cochlea, in this jelly-like substance, there are hairs that respond to different frequencies, different amplitudes, and these ear hairs, as you can see here, here's the auditory nerve, they will trigger different portions of the auditory nerve, so that way your brain processes all of the vibrations coming out of my mouth as words, and that goes back to the verbal association areas as well. Super cool. And the inner ear has two sensory organs. It has the cochlea right here for hearing, and the vestibular apparatus, which allows you to know where you're head in space and to help maintain balance, with the cerebellum and then the primary motor cortex in the precentral gyrus of your brain. Now we've talked about this. The louder the sound, the more ear hairs are stimulated. The more ear hairs. Hair cells, that's what they are. They're tiny cells that look like hairs, and they're only called hairs because they resemble hairs. They're not actually what we would consider hair, like on your head. And then the brain interprets this as it causes increased impulses and causes the brain to process this as a louder sound. Pitch activates different hair cells in the basilar membrane. The basilar membrane, anytime you see anything with the basal in it, just means the bottom. And the brain, depending on which hair cells are stimulated, this, again, labeled line code. This will trigger different neurons that process that particular pitch. And then the inner ear, this is your vestibular apparatus. We have three semicircular canals. Like I mentioned in class, they go on the Y, the X, and then the Z axis. And these help with balance. They respond for dynamic equilibrium, and we're not talking dynamic equilibrium like chemistry. We're talking about you staying upright and not feeling like you're going to vomit if you sit in a swivel chair. Anytime there's movement that causes the head to move, the fluid in the canals moves. And this pushes on the cupula and stimulates hair cells and sends messages back to your brain saying, oh, my head is sideways. Oh, my head is moving in a circle, which I'm actually doing as I'm telling you this. And the vestibule helps with balance when we are not moving. It allows us to stay upright in a chair or to stand properly. Now, smell and taste. We have olfactory receptors, and these are in your olfactory epithelium, and it's right here at the top of, this is the olfactory bulb, and the olfactory epithelium is all the way through here. There's 3 layers. They're called the nasal concha, and if you think about a conch shell, that's what they are. That's what they are formed like. They have a little bit of a curve, and the respiratory epithelium, the olfactory epithelium right here, it's covered with mucus, and as the any type of smell molecule, if you will, there's a more technical term for it, but we'll go with that. They're aromatic molecules, technical term. When they are dissolved and attached to the mucus on the olfactory epithelium, they then dissolve in and bind to the olfactory cells. You can see here, here's all the mucus. This is your skull, and then down here we have the receptors, and here are the little tiny hairs. So what happens is the molecules attach and trigger these neurons to send messages to the brain. And I studied olfaction in blowflies, and the best way that I can describe how there's all of these beautiful scents in the world and our brain can process them, it's like keys on a piano. The different keys pressed down make a different sound. And so the different olfactory neurons, the different areas of the olfactory epithelium that are stimulated, the different smell your brain will process. It's kind of awesome. And then from there, the olfactory bulbs process the information and pass it to the limbic system and the cerebral cortex. Now, if you had stuck with me through the last lecture, you'll know that the limbic system plays a large role in our emotions. And so this is why when you smell your grandmother's cookie recipe, even though you haven't seen her in a few years, you are overwhelmed with memories and images of your grandmother. Because the olfactory sense, the ability to smell, is so closely linked with our emotions and memories that sometimes scents can actually help us remember things. I have a story that I'll tell you about that in class. Now, taste buds. The tongue is a large muscle. That's really all it is. Taste buds are located on the tongue, and it's very similar to how olfaction works. So we have our saliva. You can see here in our, we have gustatory pits, and there's taste hairs. There's literally little hairs on your tongue. And these make up what we call the taste buds. And as the different molecules of whatever we eat are dissolved, they touch the different parts of the tongue. Which is why whenever you're tasting a new food, or if you're a foodie or a sommelier, you swish the food around your mouth so that it gets all the areas of what you call your palate, which essentially is just your tongue if you're talking about the sensing palate. And on your tongue, you can see all these ridges and grooves, and these are called papilla, and within them, deep in the pits, the gustatory pits right here are where we have our sensory nerves and our taste pores and our taste hairs. Anyway, so all the molecules, they attach here, and depending on where they touch on the part of the tongue, they can elicit a sweet taste, a salty taste, a sour taste, a bitter taste, or umami. And umami is more of a meaty taste, if you will. And that's it for the senses. It's been wonderful. I hope to see you next time.